Hi, and welcome to the Cell Vodcast on organelles that are involved in the processes of energy processing. And in this vodcast, we're also going to cover organelles involved in transport and structure. Remember that in addition to learning these various organelles, we're really trying to take it to the next level and understand how these various organelles are interconnected, how it is that they work together to carry out the functions that make a living cell possible. So, if we're talking about energy processing, a natural place to start is with our mitochondria. And the mitochondria is really the powerhouse of the cell. And when I say powerhouse, I mean that it is the primary organelle in generating ATP. Recall that ATP is the energy currency of a cell. It's easy for a cell to use, it's small, it breaks down very quickly, and it stores a lot of energy. So the mitochondria are taking in organic molecules like glucose. So it takes in sugar and produces ATP. In fact, from every one molecule of glucose it takes in, mitochondria can produce on average about 36 ATP. By themselves, without mitochondria present, your cells can only generate about 2 ATP from every molecule of glucose. So you get a lot more bang for your buck when you're using a mitochondria. And in fact, all eukaryotic cells have mitochondria in them. And because of that, we're able to be multicellular and carry out really diverse functions. Now remember, not all cells are created equal, so you're going to find some cells that have more mitochondria than others, such as a muscle cell, which requires a lot of energy to function. You'd find a lot of mitochondria there. Now, mitochondria are a bit quirky in that they have a few factors that you're not going to find in other organelles. For example, mitochondria have their own DNA. Okay? That is, they can replicate independently of the cell. They also have a double membrane. And strangely enough, the outer membrane of a mitochondria, which is seen kind of here, is very much like that of a eukaryotic cell. But the inner membrane is very similar in composition to that of a prokaryotic cell. It has characteristics in common. And all of these things point to, along with some other evidence, point to the idea that we know as endosymbiont theory. Meaning that mitochondria were once free living organisms, similar to a prokaryote like a bacteria, that were engulfed by another cell and instead of being digested, stayed and remained in a symbiosis, in a mutualism with that cell. They provided an advantage to that cell. They could generate a huge amount of ATP that gave the cell an advantage, and this proliferated through time. Another important part about mitochondria to remember is that all animal cells have mitochondria. And we make the mistake of thinking, okay, well, plants do photosynthesis, animal cells do cellular respiration and use mitochondria. It's important to remember that plants, while they're doing photosynthesis, that manufactures the high energy sugars. Those sugars still need to be broken down into ATP. So plants and animal cells have mitochondria. In fact, all eukaryotic cells have mitochondria, except for a few specialized ones that we'll talk about later. Next, if we're talking about energy processing, we also need to talk about chloroplasts. Now, chloroplasts are found in plant cells only. You're not going to find these in an animal cell. Okay? Chloroplasts, like mitochondria, have their own DNA. And they also have that peculiar double membrane. And they also prescribe to that endosymbiont theory, saying that they were once a free living organism that was engulfed by another one. Now, chloroplasts' main job is in harnessing solar energy. So they harness solar energy. And they convert it to high energy sugars. And what's the name of that process again? Yes, is photosynthesis. Okay? Carbon dioxide plus water plus light energy plus sun is going to yield glucose, C6H12O6, plus oxygen gas. And that can then be broken down in respiration to create ATP. Now, chloroplast belongs to this broader group called plastids. And they're not the only kind of plastid that's found in a plant cell. They're a particular one that contains chlorophyll, and that's the green pigment that's going to be involved in photosynthesis. But we also see things like leucoplasts, which store starch, and chromoplasts, which store pigments for flowers and fruit and leaves and give them their particular colors. These are really used in a couple of different features. Number one, pigments in a flower are used to attract pollinators for the plant to be able to reproduce. All right, so moving right along, we're going to talk now about some of the organelles that are involved in structural support and movement, as well as communication within the cell. This includes elements of the cytoskeleton, which is everything from microtubules and microfilaments to the extracellular matrix, which is this new understanding of this matrix of collagen fibers that's excreted beyond the cell membrane. Okay, now first let's talk about centrioles. Centrioles are found in animal cells only, and you can kind of see these here. Centrioles aid in cell division. They act like anchoring points in cell division. And they're made of microtubules, which are a particular kind of filament. And those microtubules are composed of the protein known as tubulin. Okay, we're going to see microtubules again. Their role is not just defined by centrioles, but they're also involved in producing cilia and flagella and allowing a cell to be modal and to move. The cytoskeleton is composed of 3 different kinds of filaments. The first one we'll talk about is pictured here on the left. These are our microfilaments. And microfilaments, you can see, are composed of the protein known as actin. And they have a couple of different functions within the cell that are equally important. Number one, they're involved in something that we call cytoplasmic streaming. And cytoplasmic streaming is the movement of the cytoplasm and the various organelles around the cell. We'll definitely see this when you look at amoeba, which is a kind of protist that has this kind of amoeboid, undefined shape and moves by pushing its cytoplasm against its cell membrane and then extending these pseudopods into its environment. And the microfilaments are what are helping the cytoplasmic streaming of these things. Microfilaments are also really important in muscle contraction. Okay, and allowing your muscle filaments to slide along one another and so that they can contract. The next kind of component of the cytoskeleton is here in the middle. These are intermediate filaments. And with all of these have been fluorescently dyed, so these aren't the true colors. This is showing and highlighting just that part of the cell, though. So when you look at the microfilaments over here in red, this is the microfilament stretched throughout the cell. Okay, here we see the intermediate filaments. And intermediate filaments are involved in anchoring cell parts. So we don't just want all of our organelles to be floating around willy-nilly in the cell. They're actually anchored in various places so that they can coordinate with other organelles and perform their functions. Last but not least, and the one that we'll talk about probably the most, are seen here on the far right. These are our microtubules. And microtubules, like intermediate filaments, are involved in support and help provide for the shape of the cell. But they're also involved in movement. Okay? Microtubules make up both cilia and flagella. And these are going to help cells to either be modal. In the case of flagella, it helps cells to move. In the case of cilia, sorry, 2 L's there, they're going to help the cell move materials over itself, which in the case of your lungs is going to sweep things up and over those epithelial cells in your lungs. Or in the case of a paramecium, which is this another kind of protist that looks kind of like a shoe and is covered in all these hair-like cilia. When these cilia beat like a bunch of little oars, they move water over the paramecium, which in turn pushes the paramecium through the water. These microtubules are composed of a protein that you see here called tubulin. Let's talk a little bit more about cilia and flagella, and what the difference is between the two. So, flagella are used in locomotion in single-celled organisms, and they tend to move in a whip-like fashion as you see here. Okay. Now, sperm and some protists use these. Euglena use a flagella to move around. Sperm uses a flagella to move, as well as a variety of other protists. Cilia, on the other hand, are pushing materials over the cell. And cilia are like a bunch of little oars, a bunch of fine hairs that are covering the cell itself. Okay? Your cells lining your respiratory tract have this, and it helps to sweep things out of your respiratory tract, different pollutants and mucus and things like that. But they're also found on the paramecium, as you can see in this little video clip here. Check out this paramecium. You can see those fine little projections all covering it. They look kind of fuzzy all around it. Those are all the little cilia that are beating, and you can see that it's moving itself through the water by beating all of those cilia. Okay, this is slowed down. You notice here that the time step is half time because these little critters are actually moving very quickly through the water. Let's talk a little bit more about the structure of the flagella. Now, we've said that the flagellum are made of microtubules. And we say that flagella have what we call a 9+2 structure. That is, they have 9 pairs, and you can see them here, 9 pairs of microtubules. And those pairs of microtubules make up the outer ring. In the center, we have 2 more microtubules. Okay. And they're arranged in this fashion, and they're covered by an extension of the plasma membrane. So the microtubules are projecting out of the cell, and then the plasma membrane is coming up and over them. Well, how does this thing move? And what we know is that between the microtubules, there's a protein called dynein. This is a motor protein that connects the pairs. So it connects one pair to the adjacent pair. And as it walks along, or attaches and reattaches, attaches and reattaches along one of the pairs, it's going to cause the whole flagella to flex. Now, it's all happening very quickly, but it's the dynein arms that are going to be between the 2 different pairs of microtubules that are going to attach and cause it to flex. Now, we used to think that the plasma membrane was the outermost limit of a cell. And we now know that that's not quite the case. We have beyond the cell what we call the extracellular matrix, or the ECM. And the extracellular matrix is secreted by the cell, and it's a lot of collagen fibers. And collagen is a protein. So it's these collagen fibers that are secreted out, and this is involved in support, it's involved in movement, in regulation. We see this in the white blood cell. If you haven't checked this out yet, look on YouTube. for the inner life of the cell. It was a project put forth by Harvard University and BioVisions, and it shows some really great details about how the cell works, and it shows some great images of the extracellular matrix of a white blood cell and how it's communicating with the epithelial cell of a blood vessel. So check out the inner life of a cell if you haven't seen it already. It's an awesome, awesome video. And check out the version with sound. There's also a more technical version that it describes all of the details. Now, the extracellular matrix has some other important features, like these integrin proteins here. And integrin proteins integrate. They integrate the ECM, extracellular matrix, with the plasma membrane. And proteins are able to communicate. When we cause changes in their shape, we change their function. And so by having these integrin proteins anchoring the extracellular matrix, it can also communicate information from the outside and in, and vice versa, so that the cell can communicate with its different environment. Now, all of these cells are not existing independently in your body. They're junctions, they're connected. And in animal cells, there are three types of junctions that vary in their functions. We have tight junctions, anchoring junctions, and gap junctions. And they all serve very different roles. So tight junctions, you can see here, almost look like a knit fiber. And you can see it's almost like sewing pieces together. Tight junctions seal cells. They bind cells to prevent leakage between cells. To prevent extracellular fluid from leaking between cells. Okay. Anchoring junctions, on the other hand, are going to secure cells into sheets. Okay, they do exactly what they say. They're going to anchor cells to each other. You might see these in muscle cells, so that we're anchoring these all together to create a tissue. Gap junctions are different than these two other junctions. Both these tight junctions and the anchoring junctions are securing things. Gap junctions have a totally different function. Gap junctions are working to, gap junctions seen here, they allow ions to traverse the plasma membrane. So they're allowing ions and chemical signals to spread from one cell to an adjacent cell. Now, all of these various types of junctions are found in animal cells. And animal cells, recall that we have the plasma membrane at the exterior, and we have the ECM, but we have the plasma membrane. There's nothing really rigid preventing the cell from doing other functions and connecting to other cells. This is really different than what we see in plant cells, because recall, what's on the outside of a plant cell? That's right, we have the cell wall. And that cell wall is composed of that tough polysaccharide cellulose. Okay? And that cell wall is serving to give the cell structure and shape. Okay? But we still need to allow for some amount of communication. In plant cells, we have a totally different kind of junction. In plant cells, we have something called the plasmodesmata. And what a plasmodesmata is a channel between cells. You can see over here, if we're taking a look at this plasmodesmata, you can see it here in this image, and then let's blow it up. If you're looking here, you can notice that right here where I'm tracing, this is the cell membrane. And you would expect the cell membrane to be totally separate in each cell, but in fact, the cell membrane extends and is contiguous with the cell membrane in its adjacent cell. And by doing that, and then also having this gap here in the cell wall, this is our plasmodesmata through which we can have different kinds of things flow. And this is important for communication in cells, in plant cells, and also important for transporting materials between cells. So again, you see here that this cell membrane is contiguous between the two cells, and the two cells are joined by this gap, this plasmodesmata. And the cytoplasm and materials can flow freely across both of these things. Well, these are the various structures that are involved in energy production in a cell, as well as in support and structure. I hope you found this useful. There's a great wealth of resources online for you to check out. Make sure you take a look at that Inner Life of the Cell video by Harvard and BioVisions, and keep exploring.